Every cell in our bodies, from the neurons in our brains to the skin cells on our hands, contain essentially the same instruction manual, the same DNA. However, they know that they are different cells with different functions. Well, how do they know that? Well, the answer might lie in something we call the epigenome, a second layer of instructions that sits on top of our DNA that helps to activate and silence different genes. Hi, I'm Krutik, I'm a scientist, and in today's video, I go over the epigenome and introduce this topic using a fascinating study from 2005 that used twins to identify how the epigenome can change over time. To understand epigenetics, we have to take a closer look at how DNA is organized within our cells. The famous central dogma of molecular biology is DNA makes RNA makes protein, and is a great starting point, but it hides a universe of complexity. One factor of importance is gene expression, the process of turning some genes on and leaving others off. The cells in your skin and eyes have the same genes, but have different protein compositions that are required for their respective functions. As such, these cells have different gene expression profiles, i.e., different genes are activated, and some of the genes that are activated in both cell types have different levels of activity. A lot of this is controlled by the epigenetics. Epigenetics as a whole is a set of molecular mechanisms that controls which genes are activated and which genes are silenced. And in today's video, there are two major epigenetic mechanisms we will look at. The first is histone modifications. Your DNA is not a loose string. It is spooled around barrel-like proteins called histones. These histones have tails, which can be chemically tagged. Adding or removing such chemical tags as acetyl groups can cause the DNA to either wind up tightly, hiding genes away and turning them off, or unfurl them, exposing genes and turning them on. The second mechanism is DNA methylation. This is a more direct approach. A tiny chemical tag, a methyl group, can be attached directly to specific spots on the DNA, usually a CPG site, a long stretch of DNA containing lots of C's and G's. This methylation acts as a physical lock on the gene, preventing cellular machinery from reading it. And here's the really cool part. These epigenetic markers are not fixed. They can be influenced by our age, our environment, our diet, and our life experiences. And there was no better way to prove how these epigenetic markers work other than by studying twins. One of the most immediate findings was that X-chromosome inactivation can become skewed between female pairs of twins over time. Let's unpack this because it's a beautiful piece of biology. Individuals with two X chromosomes face a potential dosage problem. Without correction, the cells would produce double the amount of protein from X-linked genes compared to individuals with just one X chromosome. Evolution's elegant solution to this is to entirely shut down one of the X chromosomes in each cell. Very early in female embryonic development, each cell independently and randomly silences one of its two X chromosomes, packing it away into a dense inactive bundle. And this turns the entire body of that female into a mosaic, a patchwork of cells where some of the X chromosomes are inherited from the mother and others are inherited from the father. The well-known example of this is the calico cat, whose patchy coat color is a direct result of the random X inactivation. Going back to this 2005 study, They found 19% of their female twin pairs had what was known as a skewed X inactivation pattern. Instead of the expected random 50-50 patchwork, where half the cells have the father's X chromosome and half the cells have the mother's X chromosome, one twin might have had a 70-30 split of active chromosomes, while her genetically identical sister had a different ratio. And therefore, the epigenetic process of X chromosome inactivation is not a heritable trait. But the more powerful proof of the effect of epigenetics on twins and all of us came in the form of epigenetic drift. The researchers zoomed in, comparing the epigenetic marks from a pair of 3-year-old twins, and compared this to marks found in a pair of 50-year-old twins. First, they measured the total amount of these epigenetic marks across the whole genome. Surprisingly, the average amount of marks was almost identical between the young and old pair of twins, but that was a red herring and did not convey the full story. The real discovery came from when they measured the difference in these marks within each pair of twins. For the 3-year-old pair of twins, the differences were tiny. Their epigenomes were virtually indistinguishable, i.e., roughly the same genes were activated in the young pair of twins. But for the 50-year-old pair of twins, the differences were enormous, and so different genes were active within the older sets of twins. This proved a concept known as epigenetic drift. Even though we start life with a certain set of epigenetic instructions, a lifetime of different choices, environments, and pure chance can cause the epigenomes of two genetically identical people to drift apart, becoming less and less similar as they age. The researchers also confirmed this across the genomes. Looking at thousands of CpG sites where methyl groups were likely to be concentrated, in the young twins these methylation maps were almost identical. In the older twins, however, these maps showed widespread scattered differences, clear evidence that their life stories had been written onto their DNA in different ways. So, we've established that epigenetic marks change with age and environment, but does it really matter? Why is this relevant to you and I? The final part of this study answered that question directly. All of these changes to histone tags and DNA methylation aren't just molecular noise, they have a real, functional consequence on which genes are being expressed by the cells. The team looked into global gene expression profiles, a snapshot of all the genes that were turned on in the cells of the twins, and that data perfectly mirrored the epigenetic findings. In the 3-year-old twin, the gene expression patterns were almost perfectly correlated, but in the 50-year-old set of twins, there were significant divergences. Hundreds of genes were expressed at different levels between them. The epigenetic drift that occurred in their lives had a real effect on their functional biology. Overall, epigenetics has fundamentally changed our understanding of what it means to have a genome. We are not simply a set of pre-programmed instructions fixed at birth. Our DNA is a dynamic script, and the epigenetic marks are an annotation on the margins, added, erased, and edited by our age, our experience, and by our environment. This wonderful study I've added to the description below uses a powerful model, identical twins, and was the first to prove at a molecular level that we are a constant conversation between the nature we inherit and the nurture we experience. Thank you so much for watching. Please like, subscribe, and share for more of this style of scientific storytelling, and see you next time for more science.